Biosensor devices make use of microorganisms or microbial enzymes. They are linked to the bioreceptors on the electrode, or known as a transducer, in order to detect specific substance from an analyte sample by converting the biological reactions to electric currents. The electric currents generated will undergo signalizations, and from there we will generate our data. Well, it's really not cheap to create one biosensor device. Thus, we must really ensure our biosensor device is a viable device. The viability of biosensor device depends on the biorecognition component, how they are immobilized strongly and successfully on the biotransducer components. By understanding the types of bioreceptors and type of biotransducer that we use in our biosensor device, we're able to know what kind of immobilization methods that plays behind them. Alright, before we start, let me clarify this. The bioelements that we are going to talk about later is the enzyme. So for the biotransducer component will be the support, or we call it a solenometric support. Alright, let's get it started. So we're going to introduce you all about 5 different types of immobilization methods.
Adsorption is a physical method of immobilization. Many substances adsorb enzyme on their surface, for example cellulose, silica gel, and also alumina. Microbial cells immobilize by adsorption by placing the cells on the porous cellulose membrane. Advantages of adsorption: no reagent is required, no cleanup step, less disruption to the enzyme. Disadvantages of adsorption: attachment is weak, hence etching of enzyme. Absorb biomaterial susceptible to environmental changes, and limited lifetime.
Hi everyone, let's move to the next immobilization method, which is microencapsulation. Microencapsulation is the method where enclosing the enzyme within spherical, semi-permeable membranes. We move to the advantages of microencapsulation method. The first one is there will be a close attachment between biomaterial and transducer, since the biomaterial has been placed behind an inert membrane. Second advantage is this method is a cheap, simple method and also very adaptable. For the disadvantages of this method are: firstly, it has weak attachment, thus leaching of enzyme from support can happen. The attachment is only by hydrogen bonding and van der Waals forces. Lastly, encapsulated biomaterial is very susceptible to environmental changes such as pH, temperature, and ionic strength.
Now, I will explain about immobilization method in biosensor technology using entrapment. It can be defined as physical enclosure of biomaterials within matrix support that are usually water-soluble polymers. It also involves covalent or non-covalent bonds between biomaterials and matrix. These are some examples of commonly used polymers in entrapment approach. Some factors that might affect entrapment include size and molecular weight of the biomaterials, nature of the polymer used, and the adjustment of pore size of polymer by concentration modification. This shows example of entrapment of enzyme by using calcium alginate. Alginate are linear polysaccharide isolated from marine brown seaweed and algae. The process involves mixing of biomaterial, which are enzymes, with sodium alginate solution and then are immersed in excess calcium chloride for hardening. The enzymes will be entrapped within calcium alginate matrix or entrapped in the calcium alginate droplet. The advantages of using entrapment are that the procedure is fast and use cheap materials, considered as simplest method of immobilization, and it involves gentle method with mild reaction condition that help to minimize loss of bioactivity. However, disadvantages of using entrapment are that large barriers created result in slow reaction of the biosensors, then leakage of enzyme through pores might happen, and they may have chance of microbial contamination.
Let's move on to the cross-linking immobilization. Cross-linking is a covalent bonding between various groups of enzymes via bifunctional agents. So, what is a bifunctional agent? Bifunctional compound links covalently to the amine groups of lysine or hydroxylysine in the protein molecules, which lead to the chemical modification of enzymes and proteins. There are two examples of commonly used bifunctional agents in cross-linking immobilization: glutaraldehyde and diazonium salt. Cross-linking immobilization achieved by intermolecular cross-linking of the protein, either to other protein molecules or to functional groups on an insoluble support matrix. Now, let's take a look at the advantages and disadvantages of this method. There are 4 advantages of cross-linking immobilization. First, simple and fast method. Second, high stability. Third, very little desorption. And the last one is enzyme strongly bound, which it can prevent from leakage. But this method also has limitations, which it can cause damage to the enzyme, expensive and poor rigidity.
Alright, we come to the last immobilization method, which is covalent bonding. So this method involves the formation of covalent bonds between the chemical groups of enzymes and the solid matrix. So take a look here. This is a solid matrix support, and we're going to bind with the chemical groups of enzymes. Here we go. Alright, but hey, Captain Rogers will be thinking that what are the chemical groups exactly that involve from the solid matrix support and from the enzymes? Alright, this is a type of the chemical groups from the enzymes that are going to bind with the chemical groups from the solid matrix support. Oh, look at them. How sweet are they? So, imagine Snitch is the enzyme, Lido is the solid matrix support. How do they bond covalently? Before our solid matrix support able to bond covalently with our enzyme, it's very important to prior activate our solid matrix support. So first activation method will involve the polyfunctional agents. So the example here we use will be the glutaraldehyde, which is the polyfunctional agents. They're going to activate our amine containing support. So this will activate our amine containing support, and from there we add in the enzymes, then only the immobilization will be successful. While second activation method will involving a peptide bonding. So there are 2 examples we're going to talk about. First is a carboxylic acid containing support. Secondly will be hydroxyl containing support. So for the carboxylic acid containing support, we'll activate it through a peptide bonding with an agent called carbodiimide. So from there, they'll activate the carboxylic containing support, and then we add in the enzymes. Or we can use the alternative peptide bonding with a hydroxyl containing support. So we can activate hydroxyl containing support with either cyanogen bromide or S-triazine. But due to the high toxicity of cyanogen bromide, we prefer to substitute it with an alternative agent. The alternative agent is ethyl chloroformate containing support. Covalent bonding has several advantages. It involves a strong linkage or bonding between enzyme to the solid matrix. There will be minimal or even no leakage of enzyme with better solubility. There will be a variety of solid matrices with different functional groups available, and covalent bonding is applied in the right application. However, there are several disadvantages in covalent bonding. The reaction must be carried out under mild conditions. It's more expensive and more complex compared to other immobilization methods. The solid matrix support needs to be activated prior to immobilization with enzymes, which is more tedious. And the chemical modification of enzymes will lead to a lost functional conformation of enzymes. Alright, that's all from us. We end this video with several applications of biosensor.